Hello and welcome to this video on the thymus and T cells. You've likely heard that the thymus is important. This is supported by various medical procedures that avoid removing the thymus at all costs. There's also various public health campaigns to try and support its presence and use. So the question is, what makes the thymus so important that countries will literally go to great efforts to try and support people and ensure that they not only have it medically, but that it continues to function as intended? And why is it such a major focus of some research into immunological function? The simple answer is that it plays an essential role in immune function. The role it plays is all down to T cells. That is T cells, not virus, so there'll be fewer, but only possibly fewer Resident Evil T virus references. Let's start with T cells, as what T cells do is the entire purpose of keeping the thyroid and why thyroid removal is a last option, even with significant problems around it. T cells are an immune cell. This should be fairly obvious so far. These are properly called leukocytes. Immune cells that play a role in defending the body. White blood cells are the common name, but leukocytes are the proper name. Leukocytes fall into two broad categories: the innate cells and the adaptive cells. Innate cells include neutrophils, macrophages, eosinophils, and more. The distinguishing feature of these innate cells is that they are not specific to any pathogen or threat, but because of this they can respond to most everything. And this means they are a jack of all trades but a master of none. T cells are part of the adaptive immune system, that being cells that can change or more accurately be selected for so they have a greater affinity or specificity to a target pathogen, such as cancer, but also bacteria, viruses, and more. This means that, as the name suggests, they adapt or become better at coping with particular problems. In order to get a T cell, first you have the pre-existing stem cells. These are part of the hematopoietic family of stem cells. They live in the bone marrow, and eventually they get a signal saying, we need more T cells. Now, typically this follows being infected by something, but it can follow other actions, let's say. To start this entire process, we begin with a signal being received by the T cell with its T cell receptor. Now, this should correspond to a peptide, a small amino acid sequence, an MHC2 or major histone compatible 2 presenting arm. Now, there are two of these. One is MHC2, one is obviously MHC1. MHC2 is only present on what are called professional antigen-presenting cells. For the purpose of this video, we can focus primarily on dendritic cells. Understand, MHC1 is more used to indicate a self rather than indicate pathogens or things that need to be targeted. This is why MHC2 is very important. The T cell, depending on which particular kind of T cell it is, if it's a CD8+ or a CD4+, which is a distinction we won't worry about in this video, will receive this peptide. And that peptide will basically tell the T cell, this is the basic stimulus that has caused us to be trying to generate more T cells. We will point out that the major distinction between MHC1 is the length of its peptides it receives. Typically they're short, somewhere between 8 and 13 amino acids, whereas MHC2 are typically longer. In some ways this is useful, as the longer an amino acid length received is, the more effectively the T cell can differentiate it from other targets, as it's a more complex key to match a more complex lock, in very rudimentary terms. But now we know that the T cells have been triggered to have an explosion in population, and this is exactly what happens. The population starts dividing. Now, initially there may only be 1 or 2 cells, and this is because we're talking about a naive T cell. Naive T cells have never been exposed to the antigen before now. There are other instances where you've been exposed to an antigen before and have a memory, and therefore this process is slightly different, but for now we'll focus solely on what happens with a naive cell. Now, these cells will begin dividing, and each member of the divided population is going to be a little bit different from each other. That difference means that they can react better or worse to a pathogen, and that's because they've all mutated just a little bit Typically speaking, most mutations are negative, so they won't be helpful. But occasionally there'll be those that are a little bit more suited towards targeting whatever the new antigen or pathogen being presented is. To know whether or not the new T cells are actually better or worse at their job, we need to have a selection process, and this is based on features, particularly that, well, one is that it won't attack the human body. This is probably called a self-reaction or autoimmune reaction, and this is a large part of what the thymus does, but we'll get to that in due course. What we need to look at for the other feature, which comes from the second signal, and it comes from co-stimulation. And get your minds out of the gutter. This is a phenomenon in which the receptors on the antigen-presenting cells are given exactly that, a stimulus to usually produce or present the products of the pathogen. So this is your antigens. Now, typically speaking, if we were to give someone a vaccine, what's in the vaccine is what the cells would be presenting, because they only need a small amount of whatever it is that they want to protect the body from. And that's because the antigen-presenting cells need to present this small amount of antigen from whatever it is you want to protect the body from to the naive T cells. Those are the T cells that aren't yet sure if they need to be selected for. In doing this, there's a particular receptor called CD28 that would take on this particular antigen, and that will tell the T cell whether or not it's meant to be kept or disposed of. This second signal, as long as it works and is actually compatible with whatever is being presented, tells the T cell that it's good to go and begin acting on whatever pathogen or other problem the human body thinks exists. In some instances, this is actually a pathogen. In other cases, it's something like cancer. And in other cases, again, it's the completely wrong target and starts attacking your own self. The reason why we have multiple steps is that the T cell receptors are a complex cascade of events, and the T cell receptor needs to be complex. The reason for this is very simple. Without that complexity, it's too easy for T cells to start doing exactly what we mentioned, which is become an autoimmune disease and attack your own body. The T cell receptor itself is also reasonably complex for this reason. It's made up of two protein chains. It needs these in order to ensure that A, it can be produced, and B, it gives that degree of complexity in a similar manner to what antibodies have in complexity. However, unlike antibodies, where there's a fairly complex structure with multiple parts to it that vary considerably, this is even more complex at the very end, but it's also somewhat more limited in what it can do just based on how large the structure is, let's say. This means that the parts that make up this structure, that is, the amino acid sequences, need to be much more widely varied. Without this, you don't get that diversity of compatibility. So now we know what T cells do, and certainly how it is that they come to be functional in terms of the T cell itself. The question is, how do you stop T cells from becoming a problem, particularly leading to autoimmune disease? This is where the thymus comes into play. The thymus is a lymphoid organ. This is just a fancy way of saying it plays a major role in the immune system. You'll find it located about halfway up your sternus and behind it, just in front of where the lungs and heart should be. T cells, as we mentioned, begin life as a precursor, that is, the hematopoietic stem cell. These were earlier described, and they undergo proliferation or an explosion in population. Once that explosion in population occurs, that is, you get the initial differentiation, basically the stem cell divides into naive T cells. Those naive T cells typically migrate to the thymus from the bone marrow. At this time, they are strictly called thymocytes, not T cells, but it's a technical distinction for most reasons. Inside the thymus, they'll undergo the process of maturation. Now, the first thing they'll do is ensure the T cells react against antigens. This is called positive selection. This obviously tells you, once they've been activated, that they're compatible with the target, and this is that selection in that population for the most effective T cells, those that are mutated, most beneficial to you. But they also have a second test, and this is negative selection, wherein they ensure that the antigens the T cell will respond to are not self. This is important, as if there's overlap between self and pathogen, and particularly using cancer as an example, you do not want those T cells, as even if you remove the cancer, they will start attacking the human body, and once they've gone out of the thymus, they're very hard to put back in the bottle. Once the T cell is matured, it will leave the thymus and it will start doing what it's meant to do, which is to deal with anything considered a pathogen, or at least a foreign body. Within the thymus, the T cell receptors have to bind to particular receptors expressed by genes within the cells of the thymus. That's because the thymocytes that fail to pass this receptor test are considered rejected, or at least failures and dropouts. Importantly, those dropouts cannot be used. This means that if they pass for whatever reason, for example, if the thymus is not working as intended, you're going to have autoimmune disease, and this is really bad. This is where the receptor within the thymus and expressed on epithelial cells within it are a selective process. This is also part of where the CD4 and CD8 designation comes into play. The number of T cells from the initial population that survive will very much vary, and this is largely down to that mutation rate. Sometimes a beneficial mutation in terms of being able to target a pathogen or antigen better also means corresponding reactivity to the self. So when you have both MHC1 and MHC2, you basically have the thymus deciding, okay, you're reacting to the target you're meant to react to, but you're also reacting to off-target, so you can't differentiate, therefore you need to be destroyed. However, that's not the only way that this can happen. There's a second pathway, so to say, in which death can occur, if you get a weak or no reaction at all to the presented antigens. This means that cells that do live, that is, they survive through this process, only get to survive if they respond enough, but not too much, to the antigens, and importantly, don't react to other antigens that they're not meant to react to. There is a small and slight caveat to this. The CD4 positive T cells that survive, that is those that are slightly self-reactive, can be retained, let's say, as T regulatory cells, and we won't go into the detail on what these are in this video, but understand their role is basically enforcers for the thymus and the rest of the body to ensure rogue T cells are dealt with. And this is why the thymus function and presence is very important. Without a thymus, you won't be able to get T cells to work well. Without good T cell function, you won't get protection from things like cancer, viruses, even bacteria to a certain extent. But it's also how T cells and the thymus play together. When it works well, everything goes as planned. When it does not, you experience immunological issues, either from a lack of T cells because, well, T cells aren't being produced to deal with a particular antigen, excess T cells, and sometimes this is even worse, as oftentimes it leads to the next issue, which is self-reactive T cells, and these cause autoimmune diseases. Or you can have instances where you're producing T cells that really aren't very good at their job, and there are more issues again that can be caused by a thymus not working as intended. In other instances, you can actually have the thymus creating complications itself. For instance, when it has to be removed due to cancer, and there are other problems again that can occur in the thymus, some of them more ironic than others. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please do post any comments, questions, or suggestions you have below.